Good morning, everyone. My name is Peter Abadir. I'm a geriatrician, a clinician scientist, and one of the PIs of the Johns Hopkins Artificial Intelligence and Technology Collaboratory for Aging Research. And it's my distinct honor and pleasure to present our keynote speaker, Professor Jianying, who, Director of the Healthcare and Life Science Research, Global Science Leader, AI for Health at IBM. Dr. Hu is an IBM Fellow and a Global Science Leader of AI for Healthcare at IBM. She's also an Adjunct Professor of Medicine at Icahn School of Medicine at Mount Sinai. Dr. Hu joined IBM in 2003 after working at Bell Labs. She has over 30 years of experience conducting and leading research on machine learning, with recent focus on AI-enabled acceleration of scientific discovery in health. Dr. Hu has served on many editorial and advisory boards, most recently on the NASEM Committee on establishing a framework for emerging science, technology, and innovation in health and medicine, and the External Advisory Board of the NIH Aim Ahead program. Dr. Hu is a fellow of the American College of Medical Informatics, International Academy of Health Science Informatics, IEEE, and the International Association of Pattern Recognition. Please join me in welcoming Dr. Hu.
Thank you so much, Peter, for the warm introduction. And I want to thank Ben and the organizers for inviting me here. I'm really delighted to be here with this amazing community and to share with you our journey of applying AI to really help with the drug discovery process. Drug discovery, as Alex very eloquently described just now, is a very long and costly process. It takes, on average, 10 to 15 years and more than $1 billion to take a drug to market. And of all the molecules that reaches the clinical trial stage, only 12% of them eventually gets approved. And furthermore, even for those few precious drugs that get approved, there remains much to be desired, particularly in terms of low precision. So in this study that was published in Nature some years ago, they looked at the top grossing 10 drugs in the US market. And what they found was for every patient that these drugs help, there are between 3 to 24 patients that they fail on. So that means an average of a very dismal, about 13% precision. So really, a lot more needs to be done. And if we come to neurodegenerative conditions, the situation is even worse, right? If you take Alzheimer's disease as an example, the challenge here is really acute because on the one hand, there has been increasing incidence of the disease. So as you see here in the US alone, there are expected to be more than 13 million Alzheimer's disease patients by 2050. And then on the other hand, there has been really a very disheartening lack of progress in terms of therapeutic development for this disease, right? So if you look at the pipeline of Alzheimer's disease medications, you see that the trial success rate in this particular area is even lower than average, is at about 2%. And if you compare 2024 to 2023, there were fewer trials that were brought forward and fewer new chemical entities. So again, this, really we need to do more. So how can AI help? Like Alex, we also believe that AI can really help, accelerate the pace of drug discovery in terms of both efficiency and effectiveness of it across the whole spectrum, the whole pipeline of this discovery process, all the way from target identification to post-approval analysis. And there are many different areas that one can put efforts on. And here we call out some concrete areas that we identified and started to work on a few years back. So starting with new molecular entity, you can use AI methods to generate new molecules and to perform property prediction to help with virtual screening. AI can also help identify new indications for existing drugs, this notion of drug repurposing, so that you can then sidestep some of the early stages of drug discovery and enhance the speed of discovery in that way. AI can also be used to help with establishing safety and efficacy profiles of molecular candidates by basically developing methods to connect observations from in vitro experiments with mechanistically linked parameters in physics-based models, right? And again, by doing that to enhance your hit rate. And AI can also be used to build models to characterize and assess the diseases, the stages of disease progression, so that you can the relevance of that for drug discovery is you can help with better target identification depending on the stage, the different factors that are at play at different stages of the disease, as well as better trial design to identify the right patients who are most likely to benefit from a particular candidate drug. And finally, better assessment of the impact of this kind of a new candidate in the context of the disease progression. And they can also be used to identify multimodal biomarkers that can, again, have an impact across the whole pipeline of disease, of drug discovery. And finally, these biomarkers combined with disease progression modeling, along with other computational tools such as digital monitoring, can be used to really enhance clinical trial design and effectiveness of these trials to allow us to better assess the true effect of a candidate on the appropriate population. So this is kind of the big picture, and what I'm going to do in today's talk is to give you some examples of the kind of AI methods, starting from the pre-Gen AI, the quote unquote more classical machine learning methods, all the way to some examples of AI foundation models that we are recently working on, and with, again, examples in neurodegenerative conditions which often accompany aging. Right. And I'll also offer some thoughts at the end on the ways to take this forward. So the first example I want to talk about is some work that we have done in disease progression modeling, again with examples in some neurodegenerative conditions. So here the idea is, just like other chronic conditions, many neurodegenerative conditions evolve over a long period of time, and there is large amount of variation in terms of what symptom you exhibit first, in what order, and for what duration. And as a result, it's very challenging to accurately assess the stage of progression for any given patient. And this is particularly true for early stages of progression, when the symptoms are more subtle and also more varied. At the same time, for the purpose of drug discovery, that early stage is precisely the stage we want to focus on, because we have more chance of actually averting that disease progression and actually cure instead of just manage the disease. So what we have done is to take advantage of the fact that now over the years we now have a lot of longitudinal data collected for many different chronic conditions, including neurodegenerative conditions, to now build machine learning models to help us really better understand how do you extract succinct characterizations of diseases at different stages of progression, and how do you characterize the patterns of that progression. And as you can see here, we collaborated over the years with many different institutions and applied this approach to many different disease areas. And this actually is one of the earlier examples of how scalable AI can be used to really support communities of discovery. We are now, taking that to the next step, leveraging foundation models to achieve better, larger scales of this kind of communities, which is something that I will touch on later on as well. Okay, so the first example that I want to talk about is Huntington's disease. So Huntington's disease is one of the most challenging neurodegenerative conditions On the one hand, it's a very simple disease to diagnose because it's caused by a single gene mutation. On the other hand, the manifestation of this disease is very complex. It has many different aspects, right? It goes across motor aspects, cognitive aspects, as well as functional aspects. And so, as we heard a lot in these 2 days, properly assessing the degeneration of even in one of these aspects can be very difficult. For example, the decline of cognition. So how do you actually combine all of these different data from all these different aspects to extract the very complex disease progression pattern is a huge challenge. And even though because it's a single gene disease and thanks to the large efforts of foundations such as CHDI, we have collected a lot of disease registry data for this condition. Past efforts in actually using that data to better understand disease progression has been very limited because they had been mostly focused on one particular aspect of the disease and very often one specific measure. And this is because, traditional statistical methods really have challenge in terms of understanding teasing out the complex patterns across these many aspects. So what we did then was to collaborate with CHDI, and then we applied our disease progression modeling framework to the data coming from 4 different registries in this disease area, and built a disease progression model that's based on hidden markup models to, for the very first time, create a end-to-end disease progression model for Huntington's disease. That aggregates all the different clinical assessments across all those different aspects that I mentioned earlier. So a few things I want to point out in this hidden markup model based what we call integrated disease progression model is that, number one, just like the modern AI foundation models, these models are also completely trained through self-learning. So there's no labeled data involved at all. And that's part of the reason why we were able to actually leverage all of those different registry data. And then, so this approach is able to automatically identify the number of states that's most appropriate based on all the longitudinal data that's observed, and the characterizations of each state in the form of conditional probability distributions of all these different clinical measures that's specific to each state. It's also able to learn automatically the transition probabilities across all of these states. And once that model is established, you can now apply that model to each individual patient, right? So given the longitudinal record, you have that patient of the sequence of clinical assessments, it's able to then assess for each one of these visit, what is the most likely state that patient is in during that visit, all the way up to the current visit. And then based on that, also project forward in terms of where that patient is likely to be in the near future, right? So this was, as I mentioned, applied to 4 different disease registries that altogether pulled together about 16,000 cases as well as 4,000 controls. And as a result, the model discovered 9 distinct disease states that spans over 4 decades of disease progression. So one thing I also want to point out here is none of those disease registries actually contain data that covers 4 decades. It's different registries tend to focus on different stages of the progression, but because of the probabilistic nature of this hidden markup model-based approach, we're able to essentially stitch together these different registries and come up with this end-to-end disease model that really covers the different stages of progression, going all the way from prodromal to what the clinicians refer to as transition stages, and then as well as manifest stages. So some examples that you see here in terms of how this model is able to pick up some of the more complex progression patterns is you can see at the bottom there's the plot of the different clinical assessments from motor domain to cognitive to functional, right? So if you look at compare state 4 and 5, for example, these are 2 states in the transition stage of the disease. then you can see there isn't much change across all the motor assessments as well as the cognitive assessments. But if you look at the functional assessments, you see a steep decline almost across board for all the functional assessments. So that points to that for most patients, they will experience a steep functional decline right before they transition into the more manifest stage of the progression. And that's something that's very interesting to the clinicians as well as to the pharma companies who are developing therapies who want to catch these patients before they transition into the more severe states. And then on the lower right-hand corner is one example of what I mentioned, where you apply this model to an individual patient, that provides the physicians with a more nuanced understanding of how this patient has been progressing and where the patient is at, right? So this is one particular patient that was tracked for a long period of time, almost 15 years. Now, if you look to the very right column, this is the standard clinical staging. And you can see that was used at the time, right? And you can see the patient was prodromal for many, many years and then suddenly jumped to first stage of HD stage 1 and stay there. But if you apply our model, the integrated disease progression model to it, you see the patient, yes, started with stage 2, which is a very early pre-symptom state. But then at some point, the patient switched to stage 3 and 4, meaning it's about, the patient is about to hit the more symptomatic stage and then finally got to stage 4, which is one of the manifest states. So again, this gives more insights, earlier insights to the physicians when a patient is, about to progress to the more serious stage, and so that, everyone, including the caregivers, can be more prepared. Okay, so, and those results was published in Movement Disorders 2 years ago, and this is just one more recent paper that we published, building on that work, applying the disease progression model to a larger population now to really carefully assess the discriminative power of different clinical assessments across the different stages of the progression. And what we showed was that, and this was just published at the end of last year, what we showed was that the discriminative power of these different clinical assessments are very heterogeneous. Not surprisingly, some are more effective at different stages of progression. So, for example, motor symptoms, particularly total motor score, have the highest discriminative power between the very early stages of disease progression into the transition stages, whereas cognitive assessment has good discriminative power across the progression spectrum, except for between the very early 2 stages of progression. Again, what this does is it really provides insights into not just patient care, but also clinical trial design in terms of what are the appropriate endpoints to use for a therapy that's targeting different stages of the progression. Right. So another example I want to highlight is that same type of disease progression modeling approach now applied to Parkinson's disease. It's another very complex neurodegenerative condition. Similar to Huntington's disease, it has very heterogeneous progression pathways. And so if you just use a summary score as a measure of disease progression, you are likely to be masking a lot of more complex progression patterns. So here again, we applied the hidden marker model based approach, but there is one additional challenge here in Parkinson's disease compared to Huntington's disease is, unlike in the case of Huntington's disease, for Parkinson's, there are actually therapies that are very effective in controlling some of the symptoms for some of the patients. And what that means is this could actually end up masking the true disease state of the patient if you use a very straightforward approach to just build a model around the clinical assessments, right? Because one patient could respond very well to a medication that controls tremor, whereas another patient may not respond well, even though these two patients are both at the same stage of the disease progression. So to address this challenge, we created another variation of hidden Markov models, which is called input-output hidden Markov models. that allowed us to build into this model the capability of adjusting for effect of medication. And that adjustment can be done on a state-by-state basis, understanding that the effect of medication can be different at different states of the progression. And it's also personalized, meaning the effect can be different from individual to individual, right? So we applied this model to the largest Parkinson's disease data, that's PPMI, that's collected by the Michael J. Fox Foundation, which is the foundation that we collaborated with in this work, and identified, in this case, 8 disease states with very nonlinear progression patterns, which we published in 2021 in Nature Communications. And again, it unsurfaced a lot of interesting progression patterns that can lead to help with not just patient management, but also in terms of trial design and hypotheses for potential new mechanisms and new targets. All right, so the next example I want to talk about is causal inference for the purpose of drug repurposing. So the goal here is to identify drugs that have a desired effect on a outcome that is not originally indicated for that drug, right? So that's why this notion of repurposing. Now, the challenge here is if you just compare treated versus untreated cohorts from observational data, you are going to get a lot of false positives that's caused by confounding factors. And these confounding factors are inevitable when you try to analyze observational data, which is very different from carefully designed clinical trials data, right? So I was very happy to hear Alex talking about this dual purpose clinical trial where you can, at trial time, be conscious of a different indication that you're collecting as well. But even in that case, the challenge remains that what you are very carefully balancing for one outcome in your trial design may not be so well balanced for your second outcome that you also want to study. And so even in that context, some degree of causal inference is needed in order to make sure that the data you collect actually is analyzed properly for that extra indication, right? So there has been a lot of work in developing different causal inference methods that's aimed to adjust for this kind of bias in the data and confounding the data. But the process is typically very ad hoc and takes a long time to design for any particular study that you want to carry out. So what we did was to create a suite of AI tools to make this process more scalable and more streamlined. And so here is one example where we applied it, in this case, in collaboration with Teva, to apply this causal inference on a large data set for Parkinson's disease patients, but with the purpose of identifying potential drugs that can be repurposed for, addressing the onset of dementia in that particular population. And as you see, we were able to identify some promising candidates. And so the causal inference engine that we developed for this purpose has been open source. And as you can see from the numbers, has spawned a very, very active open research community that continues to build on this and expand on this work. And more recently, in collaboration with the Cleveland Clinic, we then further integrated this causal inference engine into a larger framework for repurposing in the space of for Alzheimer's disease. So here we first used a type of AI model called variational autoencoders to generate joint embeddings from 3 different data sets with single-cell RNA sequence data that covers 5 different regions in the brain. We then performed trajectory analysis on the different cell types that have been identified using those embeddings, and then followed by transition network analysis on protein-protein interaction networks to generate hypotheses for potential drugs that can be repurposed for Alzheimer's disease. And then these hypotheses were cross-checked on real-world data using causal inference to check whether they actually indeed have a beneficial effect on Alzheimer's disease based on the real world data. And this was the result from this study was published at the end of last year. And as you see, one candidate emerged as through this whole exercise emerged as a particularly strong candidate, and that's ketorolac. Okay, so these are the examples of in recent years how machine learning methods can be applied to help with different tasks in drug discovery. And the question is, as someone already raised, in the new new age of generative AI with this new wave of technologies, how do we leverage that to take it to the next level? Right. So, as we have all witnessed in the last couple of years, it's really clear, and as was discussed in the previous panel as well, that this new wave of AI technologies in the form of AI foundation models or generative AI is going to have really profound impact on almost all aspects of human endeavor. And one area that has attracted really intense investment and effort is the area of biomedical research and for the purpose of drug discovery. So if we step back a bit, what is a really meaningful and ambitious goal for us to achieve here with the help of foundation model is this notion of building multi-scale representations that would allow us to then drive all sorts of in silico studies, from hypothesis generation to virtual experiments to simulations, so that we can achieve better understanding of the biological interaction at all relevant scales, from molecules to cells to tissues, so that we can identify novel drugs and more effective candidates. And this is a grand vision that has been achieved, has been attempted at before. What's different now is really this new class of AI methods called AI foundation models, and so it makes sense for us to say the first step now is to then look at what really can be done with these AI foundation models, right? And so as we all know, a key characteristic of AI foundation models that allow them to be this powerful is the fact that through these methods, you can now learn from large amount of unlabeled data through self-supervised learning, which can then arrive at representations that seems to be able to capture the intrinsic entities and relationships from that data, which can then be used to drive many, many different downstream tasks. And so applying that to biomedical space, we have been building these biomedical foundation models that takes input from large amount of multimodal data that's relevant for drug discovery, and then use that to fine-tune specific machine learning models for different tasks in the drug discovery process, going from target identification to the generation and property prediction for small molecules as well as biologics. And if you put that in the context of that drug discovery pipeline, what have been trying to do is to again reach that rich representation that you see in the middle by combining all these multimodal cross-domain data through different methods, going from early fusion to mid fusion to late fusion, and also trained with many different downstream tasks in drug discovery in mind, so that these representations can then be used to fine-tune for those tasks and to perform de novo generation as well as computational screening to reduce the need for the very expensive and time-consuming wet lab experiments that Alex mentioned earlier. So to give you, just very quickly, obviously we don't do this alone. We work with many different partners to be able to validate and scale our biomedical foundation models. We are also very, very mindful of the need for open innovation in this space. So to that effect, we worked with many partners to create this AI for drug discovery working group, within the AI Alliance, which we kicked off at the end of last year with a Open Innovation Forum here in Boston. As you can see, we had really wide participation across government, industry, and pharma companies, and we are following up on that with specific projects that are focusing on benchmarking as well as better understanding of the multiomics, multimodal space in terms of their impact on, what type of models we should create for cancer. And as you can see, also, we started that effort with 2 open sourced models out of the AI Foundation model portfolio that we have created. And already we are seeing a lot of people using these models and fine-tuning it to their different tasks. And for anyone who is interested, I encourage you to check out the webpage for this working group and participate in these projects that are going on. And very quickly, a couple examples from this. Now, we have a really suite of models from this family of biomedical foundation models. And as you can see on the right-hand side, that has been trained on large amount of publicly available data, going from multiomics to proteins to molecules. And a couple of very, very quick summaries. In the omics space, we built both what we refer to as DNA-aware, SNP-aware DNA models to help us, uncover new gene and disease associations, as well as single-cell RNA sequence models that help us to address tasks such as cell type annotation, gene perturbation prediction, all very important in uncovering potential new targets. And then another example I want to talk about, I'm going to skip this one, is in the small molecule space. We have also been really looking at how to capture structure, which we believe is really the key in order to generate the right representations that can be used to for the purpose of both generation of new molecular candidates as well as property prediction for downstream tasks. And one approach of being able to capture the rich structure information is by looking at incorporating different views of the molecule, right? So if you look at the next page, we created this approach called MELON, which is multi-view embedding with late fusion, where we look at the different views of molecule. That's image view, the sequence view were represented as smiles, as well as the 2D graphs that really look at the interactions between different atoms within the molecule. We created different embeddings for each one of these views and then added another attention layer on top of this to look at how to combine these different views to come up with a more a richer structural representation of the molecule. And that attention layer, these ways can be further fine-tuned for any downstream tasks, because as we heard a lot early on, just having that foundation model by itself doesn't necessarily mean you're going to get good results for your downstream tasks. That fine-tuning step is really important. And one more thing I want to point out is that in training most of the large language models, the standard method in the pre-training stage is the standard mask language model, right? You take out something from the sentence and you ask the model to fill it in. But when you look at these, when it comes to molecules and these different views of molecules, there are many, many different ways for that pre-training that are much more effective than the standard method of pre-training. So we also developed many different methods that's specific to each one of these specific views to make that pre-training approach more effective, all with the goal of eventually getting at the representation that really captures more of the structural information within the molecule. And so we tested it on a large number of downstream tasks, again, going over many different benchmark data that's available out there, and validated across tasks that goes from ligand protein binding to molecular solubility to toxicity to metabolism, and demonstrated that this approach of combining these different views really gives you very robust performance across all of these tasks whereas any single view, as you can see, would perform better in one and not so good in the other, right? So that again shows this notion of fusing different elements and different views really does enhance the robustness of the base model across these different tasks. And then again, collaborating with the Cleveland Clinic, we worked on applying this foundation model to the specific study of virtual screening for the purpose of identifying candidates for Alzheimer's disease, right? So in this case, we took our base model and fine-tuned it on experimental binding affinity data for 106 GPCR targets. As you can see on the left-hand side, we achieved really good performance. And then we zoomed in to 33 out of these targets that have been identified in previous studies as being related to Alzheimer's disease. And then using these the models for binding affinity, the fine-tuned models for binding affinity prediction, we then screened over 500 metabolites as well as over 2,000 FDA-approved drugs for strong binders to these AD-relevant targets for the purpose of identifying candidates that may have an effect on Alzheimer's. And what you see here are two targets that surfaced with strong binders, one metabolite, one FDA-approved approved drug. And for each strong candidate, we then did a docking study to understand what might be the binding site. And for one of them, we actually uncovered a very non-classical binding site, which could lead to, discovery of new potential mechanisms. And by analyzing the attention weights in the embedding space, you can then identify substructures of the molecule that may contribute to that strong binding. And again, you can look at it both from the image view and the graph view and get complementary information that way. So I think I need to wrap up, but just one last note is, yes, it's important to build these domain-specific foundation models, but just having a library of domain-specific foundation models is necessary, but not sufficient, right? So as these large language models are gaining more and more strong capabilities, particularly as the reasoning capabilities start to emerge, there has been a lot of recent focus on this notion of agentic AI, which is the idea of using large language models as the reasoning engine to basically determine what task to perform in what order with what data input, right? So this really is pointing the way to this notion of AI scientist, of having AI models, not just a single model, but what we refer to as compound AI. So combination of large language models combined with domain-specific foundation models together with physics-based models and other computational tools, but together with the human scientist as backup to really accelerate this process of hypothesis generation, knowledge aggregation, and leading to new discoveries. So to conclude my last slide, it's really, really exciting time that we are at, and I believe we're really at the point where we can very soon realize this vision that was described in this paper by Cheng and others in Cell Reports Medicine a couple of years ago, of really creating a end-to-end AI-enhanced drug discovery system that pulls in all the multimodal data and that combines all the machine learning models, going again from large language models to domain-specific foundation models to physics-based models, to really accelerate this end-to-end workflow, right? All the way from the identification of disease subphenotypes and hypothesis of new targets to de novo drug design and virtual screening, and to response prediction and enhancements of clinical trials, which, as Alex said, is the last, but very time-consuming step of drug discovery. So with that, thank you very much.